Hello and welcome to Chapter 5. Chapter 5 is going to be all about viruses from taxonomy, structure, morphology, replication, infection type, cultivation, as well as how viruses affect human health. This is going to be a pretty hefty chapter, but it's also the last chapter of Unit 1. So let's go ahead and dive right in. First and foremost, if you did not know this already, viruses are extremely abundant. You are kind of swimming through an entire world of viruses. It is ridiculous how many there are. Not just viruses in general, like they're different types and different things, but in general, their quantity. So, we are still learning how viruses have shaped our planet as well as its organisms. We do have kind of an interesting thing to talk about on the next slide, but what I want to get across here is that it's honestly really amazing that we aren't always sick. But there is a primary reason why we aren't, and that is our immune system. So we have a way to fight off infections, but that is not true for everybody. Some people are immune compromised, or they have a lowered immune system, or they haven't built an immune system yet, like in infants. So, kind of backtracking a little bit to that last slide, when it comes to the way viruses have shaped the way that other organisms now exist, it's important to note that when viruses infect a host cell, they can affect the DNA of that host cell. Specifically, when we talk about bacteriophages, we're going to talk about some really interesting features of what a bacteriophage does to bacterial cells. And then this will make a little bit more sense. This 10 to 20% of bacterial DNA containing viral sequences. So kind of hold on to that for just a minute in your head. And then when we get to the different types of viruses, that will make a little bit more sense, probably in this second part of Chapter 5. But humans are no exception to this either. We have about 8% of our genome consisting of sequences that come from viruses. That is how involved viruses have become in our cells. Remember that viruses are not cells themselves. They are acellular, meaning they lack a cell structure, and they are classified as obligate intracellular parasites, meaning they have to have a host cell. So this is going to be very important when we talk about their reproduction, as well as a little bit of a debate here coming up soon as to whether or not viruses are alive. When it comes to virus size as well, it is important to remember that when we talk about that size, we are talking about nanometers. So remember that is that particularly small size, even smaller than bacteria. They are also very simple little acellular structures, okay? Because they do not have that many genes. Again, they don't have cell structures. And when we talk about what makes up a virus, at least at the base level, they're only composed of two parts. So kind of hold on to that for a minute here too. When we get into what makes up a virus, again, they're only made up of two parts. They are very, very simple. We have a few examples here of various viruses and their sizes comparatively to an E. coli bacterium. So that is that big pill-shaped structure at the bottom of the screen compared to things like a bacteriophage, rhinovirus, rabies, chlamydia. Ebola is actually a pretty large virus comparatively to the other viruses. And then you compare all of that to just a very small section of a human red blood cell, which is a eukaryotic cell, compared to these acellular structures, as well as a prokaryote. The size differences are really kind of jarring almost, how small a virus can be. Moving on to the virus debate, there are a handful of characteristics that define a species as being a living organism or not. And viruses really toe the line between yes and no. Two of the biggest things that go against viruses being a living organism is that they are not cells. And in order to be considered a living species is you need to be one or more cells. And if you already don't meet that requirement, well, you're probably not a living organism. From there, you also have to be able to reproduce on your own, and viruses cannot. They need to have a host cell and the host cell's DNA in order to reproduce. So those are the two big features of whether or not a virus is a living organism or not. My personal opinion, I think it's pretty clear viruses are not living organisms, but that is not going to be on the exam. That is more so just my opinion. Instead of saying that a virus is alive or dead, we are going to say that viruses are active or inactive. So an active virus is going to be active, of course. Inactive means that it is no longer able to infect and then reproduce in a host cell. So much like classifying animals and bacteria and plants and all kinds of living organisms, we still need to be able to classify viruses. It's just a little bit different than how we classify other organisms, especially ones that are living. Viral taxonomy is going to be primarily focused on what is the host that can be infected by that virus, and then what is the disease or diseases that they can cause. So a couple other classification systems will emphasize what we just mentioned, as well as the structure of the virus, the chemical composition, and similarities in genetic makeup. Now, prior to more modern technology, we wouldn't have been able to do most of that classification. So kind of keep that in mind. This is a little bit newer of a classification for the reason of technology, our ability to be able to compare viruses now that we can analyze them properly. You do not need to know the number of orders, families, and genera, but you do need to know the order name, family name, and genus name when it comes to viruses. So we have Virales, Viridae, and Virus. There are a decent amount of viral species, and we're still discovering more viruses every day, which is kind of crazy, but makes sense. Again, that technology is finally there. We are making connections between diseases and afflictions, conditions, etc., between viruses and human health. There is also an important thing that we need to talk about, and that is that within viruses, there's also subspecies which we call strains. So when it comes to something like herpes, there are strains of herpes that will have different names. However, we will not call a virus by its genus and species name like we do with bacteria. That's an important thing to know. We just call it by those different strain names. So we've been talking about how viruses need specific host. Now, this is kind of a weird concept to think about, but not all viruses can infect all kinds of living organisms. You have to have a specific type of virus that infects a specific type of host. And more than that, they have to be able to, or they will have the ability to infect that host specific cells. So what I mean by that is something like feline leukemia cannot infect humans. Okay, feline leukemia is a virus that only infects cats and specific cells of cats. Okay, another example is hepatitis. Hepatitis is going to infect humans, that is the host, but specifically in the host, it can only affect liver cells. So you see how it has to be a host, but also specific cells within the host. Okay, so viruses are going to have, in a sense, kind of for lack of a better word, a key for specific doors. These doors are going to be receptors on the host cell. The virus has to be able to chemically bind to those specific doors, those receptors, and be able to unlock those doors. Now, the way that viruses move, and they don't move consciously because they are very simple. They do not have a way to navigate through the environment. It is by chance. So if by chance that virus comes into contact with the host, and then by chance within the host gets to the cell that they are able to unlock that door and get into the cell, that is how a virus causes an infection. But it's very important to note that viruses are doing this perfectly by chance. Moving on to viral structures, kind of piggybacking onto that idea that viruses are incredibly simple organisms. So they are going to have two parts to making a virus, and that is going to be a covering and a central core that come in a capsid as the covering, must have, that is a must have, and a nucleic acid molecule as that central core. That is a must have. So you have to have those two things in order to be a virus. There are extra things that can happen and be added, and that's perfectly fine. But in order to be a complete viral particle, you have to have a capsid and a nucleic acid molecule that comes in the form of a DNA or an RNA molecule. Okay, when these come together, they create that complete viral particle. We call that a viron. Okay. We will talk about those extra structures, extra pieces, if you want to think of it like that, eventually, but we are going to focus on these two required structures to make a virus. So make sure for this slide you know that a capsid and nucleic acid are absolutely required to make a virus. Must have. This is base level. So the capsid is going to be a protein coat or shell surrounding the virus. Okay, it's going to be made up of proteins called capsomeres. The nucleic acid is going to be inside of the capsid, and this is going to come in the form of DNA or RNA. Now, what's going to be very odd to talk about next is that the DNA or RNA can come in a few different options, but it will only be one of the options. There are 6. Okay, so there are 6 possible options for the nucleic acid that is a must-have for a virus to come in. So let's talk about that. All right, here are those 6 options for nucleic acids. We're going to start with the DNA options. So there are 2 possibilities for DNA. Remember that DNA is normally double-stranded. That is what we have in our DNA. That is what prokaryotes have in their DNA. And that is a possibility for viruses. That is that first possibility. The second is single-stranded DNA, which is crazy to think about because for us, if DNA is single-stranded, that means it's broken. So a little bit odd. Remember that when it comes to the bases in DNA, we're talking about A's, T's, C's, and G's. This is still the same for viruses as it is for eukaryotes and prokaryotes. Now, when we look at RNA, this is where things get a little bit funky and weird. So remember that when it comes to RNA, it is normally single-stranded. But for viruses, the next possible option of the 6 is double-stranded RNA. So What does that mean? That means it's our A's, U's, C's, and G's, but double-stranded, which is kind of crazy to think about. The next two options for our nucleic acids in viruses are positive sense single-stranded RNA, negative sense single-stranded RNA. So those are the two next options of RNA. What this means is that fourth option, the positive sense single-stranded RNA, is like RNA classic. It's the RNA that you think of when you think of RNA. It is single-stranded RNA, A's, U's, C's, and G's, and it is going to make proteins. Easy peasy lemon squeezy. For the negative sense single-stranded RNA, this is where things get really weird. What happens is in that DNA to RNA to proteins, instead of going from RNA to proteins, there's some intermediate stage between RNA and proteins that we don't really know enough about, but we know that something is happening, which is why we call it negative sense, and that's that extra step. So we call it negative sense, but it's still single-stranded and it's still RNA. There's just something extra happening in that process of RNA to proteins. We just don't know what it is, but there is something there. So that is up to 5 of our nucleic acids. Moving on to the 6th, this is retroviruses. These guys are pretty neat because it is still RNA, but this is where we're going to start with RNA, go to DNA, back to RNA, then to proteins. So it's like going backwards first and then forwards into the proteins. This is only found in retroviruses. So it is single-stranded RNA. It is still RNA, but it is that backwards then forwards retro situation. So just to recap, our 6 options of nucleic acids: double-stranded DNA, single-stranded DNA, double-stranded RNA, positive sense single-stranded RNA, negative sense single-stranded RNA, and retroviruses. Make sure you know all of them. Guys, I never want you to remember any examples unless I tell you to remember examples. This is not a slide you have to remember. It's more just for your viewing pleasure as far as what type of virus is going to have those different examples of nucleic acids that we just talked about. So just for your viewing pleasure, just for fun. All right, so we have covered those 2 very important structures that create that nucleocapsid. Those are the important parts of a virus, the must-haves, the capsid and the nucleic acid. From here, we're moving on to viral structures that could be on a virus. Not always, don't have to have them, but they could have them. So the first is an envelope, and not all viruses are going to have them. This is a lipid bilayer covering the nucleocapsid that is obtained, make sure you know this, from the host cell. So we will talk about this here in just a second. But again, it is something that the virus steals from their host cell. What some viruses that can have an envelope or not contain are spikes. These are the structures that you can see on these 2 viruses that we have in front of us here. These are called spikes. This is going to help the virus attach to the host cell, and we're going to talk about that in a minute. Make sure that you know that the virus on the left is a naked virus. The virus on the right has an envelope, so it is an enveloped virus. It has that extra layer. It is wearing that layer. So it is enveloped versus naked. So let's talk about these enveloped viruses a little bit more. What you can think of it as is the virus stealing the plasma membrane, or the membrane in general, of the host cell to wear on the outside of the virus. Kind of like a terrifying little skin poncho. This is going to help the virus do a few things. It can help the virus hide from the immune system of the host, not just the host cell, but of the host. So the immune system is not going to be able to recognize the virus because it's wearing a skin poncho of the cells that it just infected. So kind of like a mask, if you will, or a disguise. It can walk right through other membranes of other cells in order to infect them as well, because it's wearing that plasma membrane. The other cells that it can infect are just going to fuse with that plasma membrane and just let it right in. So it's kind of terrifying that some viruses are able to use this ability, this enveloped virus technique, or you can think of it, as I said before, as a skin poncho. Again, serves kind of two purposes. It fuses with the plasma membrane of other cells and allows the virus to hide, kind of like a wolf in sheep's clothing. More on viral structure. So this will be on the inside of the virus. We can have enzymes. Remember that enzymes are going to be proteins. These are a catalyst for a special job of some kind. And anything that ends typically, I will say that, typically anything that ends in ACE, ASE, is going to be an enzyme, meaning it has a special function. Okay, we have three enzymes that I need you to know. We have polymerases. This is going to synthesize DNA and RNA. Remember that synthesize means it's going to make something. We have replicases, which replicase sounds like replicate, meaning it's going to copy something, and that thing that it's going to copy is RNA. And we have reverse transcriptase. This is going to synthesize DNA from RNA. And I know that sounds very similar to polymerase, but make sure you're seeing the difference there. Polymerase is going to make DNA and RNA, whereas reverse transcriptase is going to make DNA from RNA. Very important. Where you're going to see reverse transcriptase is in retroviruses, which makes sense based on what we were talking about before with those different types of nucleic acids that you can find in a virus. Now, for the most part, our cells are going to do a lot of the work for the virus, but in some cases, we don't have what the virus needs to perform the function that it wants to do. So the virus is going to have to bring what they need to perform certain functions. It's kind of like if you had a very special piece of cooking equipment, like a wok, and you go into someone else's kitchen saying, I'm going to make you dinner, and they don't have a wok. You're going to use their kitchen, but you bring your own wok because you know that that's going to work the best in whatever you're making, if you get what I'm saying here. We do want to talk about the capsid arrangement and how it's going to create the morphology of our viruses. There are three flavors of morphology that I want you to know. This is the first option. This is helical. It is a tightly wound coil, kind of resembles a spring. We're going to see this as our first option. This is our second morphological option. Also, again, still based on our capsid arrangement. All of these are based on the capsid arrangement. This is icosahedral, which not good at saying certain things sometimes, but hey, that's me. This is going to be made up of equilateral triangles and arranged in a symmetrical shape. So this is kind of a really interesting looking morphology as far as a virus goes, because it's all based on geometrical patterns. And then last but not least is going to be our complex morphology. It is the combination of options 1 and 2. This is going to be what we find in bacteriophages. Remember, we talked about bacteriophages before when we talked about horizontal gene transfer and transduction. So these guys are going to be those little spidery creatures as far as viruses go that inject into the cell of the bacteria. But they are a combination of that helical and icosahedral shapes for viruses. Because bacteriophages are just so darn cool, we got to take another look at them. So here they are in action. On the left, we have a virus that is far gone. We have those bacteriophages in the blue that are on the outside of the cell as well as inside of the cell replicating, making more viruses. That cell, that bacterial cell is gone. It is going to die. On the right, that is just the beginning of this cell's nightmare. We have those little blue spidery guys on the outside of the cell, probably an E. coli. And they are probably in the process of injecting their nucleic acid into the cell in order to start getting to work on replicating and doing all kinds of chaos and havoc for that cell. So there are multiple pathways for how a virus can replicate. We're going to talk about a few of them here, but this is going to be one of the main ways that a virus is going to be able to replicate. We're going to call it the replication cycle. Sometimes this is called the life cycle of a virus, but we got to be kind of careful with that because, again, remember that viruses are not alive. They are active or inactive. They are not living organisms, so it's a little bit misleading of a term. So let's stick to calling it the viral replication cycle, but just know that it's kind of synonymous with calling it the life cycle. And we are specifically looking at animal viruses. This is going to come in the form of 6 steps. So adsorption, penetration, uncoating, there is an asterisk next to it because of reasons. It is an optional step. It just depends on the penetration step. We'll talk about that. Synthesis, assembly, and release. What's important to note is that the whole process, these 6 steps, can take anywhere from 8 hours to 36 hours, but in reality, that is such a short amount of time for thousands of new viruses, thousands, to be made. So this whole process, while yes, it is kind of a long, lengthy process, it's only taking hours. And even if it takes that longer amount of time of 36 hours, that's still a very short amount of time for a virus to replicate thousands of new virus particles. All right, starting with step number 1, adsorption. This is going to have a lot to depend on the host range, which we talked a little bit about before. So it's going to be very specific for every virus, whether or not they actually make it to the host, whether or not once in the host, they make it to the host cell that they're able to infect. And if they get to that host cell, does the virus itself have the key that it's able to unlock those receptors and get into the cell? So what we call this is tropism. These are specificities of viruses for certain tissues. You can think of it like how picky is a virus, okay? If they have a higher tropism, then it is less likely the virus is going to be able to infect that cell. If they have a lower tropism, that means that they are less picky, they can infect more hosts, and they're less picky about the types of cells that they can infect. It's also very important to remember again, we kind of talked about this before with host range, that viruses do not make active decisions in their movement and where they go. So for a virus to infect a host and then a host cell, it's going to be a random event. So viruses, by chance, are being moved by the universe and randomly bump into that host cell. And if they're able to unlock those receptors and get into the cell to cause that infection, fantastic for them. That's great. But more often than not, that virus is bumping into things that it cannot infect, cannot do anything with, cannot do anything about, and that is just the odds. It really is kind of the stars have to align in order for an infection to occur. It is very interesting. We have a very simplified view of a virus infecting a host cell. So this is a term called docking, where the virus is using those enveloped spikes to connect to those receptors of the host cell and is potentially being let into the cell in this first step of adsorption. All right, we are on to step 2. This is penetration. You can think of it like, well, how does the virus get in? So, in order for a virus to be able to penetrate the host cell, there are 3 different methods that the virus can use. The first is endocytosis. I want you to try to remember back to early A&P when we talked about cells, where endocytosis means into the cell. All right, this is that process for a cell to envelop a particle, okay, using the plasma membrane. Now, what's going to happen is that the virus is going to trick the cell into letting it in. And this is usually by using those receptors and a chemical. It has essentially the right key to the door, if you want to think of it that way. So the virus is tricking the cell to opening the door and then letting it in using the plasma membrane of the cell itself. This is not the same as the enveloped virus. This is more like kind of a Trojan horse situation where the virus is knocking at the door and the cell is letting the virus in, not realizing that it is not a good thing to let into it. Membrane fusion is going to be where the virus has that enveloped lipid bilayer surrounding the virus, that skin poncho. And what happens is the cell and the virus, the envelope of the virus, melt together or mesh together, and the virus is then let in that way. The last makes a lot more sense until we have like an image, and that is injection. And that is what bacteriophages will typically do, where the virus is going to essentially, like a syringe, stab into the cell membrane and then inject the nucleic acid or genetic material into the host cell. Again, bacteriophages will do this. We've kind of seen images of this before. So let's look at some images of endocytosis and membrane fusion and talk through that, because it's one thing to talk about it, it's another thing to see it. All right, so we are looking at those 2 processes of penetration. On the left, we have endocytosis. Remember, that's going to be like ringing the doorbell, and the host cell is allowing the virus to come in. How this is going to happen is the virus is kind of ringing the doorbell in the sense of touching those receptors using the spikes or chemicals on the outside of the virus itself. From there, the cell is being tricked if you want to think of it that way, into allowing the virus to come in. And how this happens is the cell is going to invaginate and create something called a vesicle that envelops the virus and creates essentially like a bubble using the plasma membrane. And we can see that there where the virus is now surrounded by the plasma membrane, but it's in a vesicle, and it then must bust out of that plasma membrane vesicle and release its nucleic acid and then cause the infection. On the right, we have the membrane fusion type of penetration, where the virus is wearing that skin poncho, tricking the cell, also kind of a trick, into fusing with the outside of the virus so that they mesh or fuse together, and then it just releases the virus directly into the host cell. The difference between these two, and there's kind of a big difference here, is who's doing all of the work in order to get the virus into the host cell. The endocytosis process, the host cell is doing all the work. It is opening the door, if you will, allowing the virus to come in and pulling the virus in. For membrane fusion, the virus is the one having to do all the work. It's wearing the skin poncho. It has to fuse with the plasma membrane of the host cell. And then from there, it's going to release itself into the cell. So the big difference is who's doing the work. And of course, we've got our lovely imagery of injection, but I don't feel like this one was so much a problem to try to imagine. We've got our bacteriophage coming in, attaching to the outside of the cell. That's very important to remember that when it comes to bacteriophages, they stay on the outside of the cell, and then they are going to use various structures. You don't have to know the whole process. We don't really care. Where they're just going to inject past that plasma membrane like a syringe and inject their nucleic acid into the host cell. So they don't actually put themselves into the host cell. They're just depositing the important information, the important material into the host cell and then, taken off. Now, step 3 should make a lot of sense as far as step 2 goes. This is optional because it is an uncoating of the protein coat, the capsid. Okay, so the outside of the virus needs to be removed in order for the nucleic acid to be released in the host cell. If the virus is inside of the host cell, such as through the process of endocytosis and membrane fusion, then yes, we need to go through uncoating. You need to take off that little protein coat. If, however, you are a bacteriophage and all you do is attach to the outside of the host cell and you inject that nucleic acid into the host cell, well, there's no reason to uncoat because you've already released your nucleic acid. There's no reason. So that process of uncoating is going to be different for endocytosis and membrane fusion. But the big thing here that I want you to kind of understand is that if the virus goes through endocytosis or membrane fusion, step 3 must happen. If the virus is a bacteriophage and it uses injection in order to get into the host cell, then step 3 is just optional. Well, it's unnecessary, not optional. It's unnecessary. Step 4 is synthesis. Remember that this is going to be that making process. So in making things, what we're talking about is we're either making DNA, RNA, or when it comes to retroviruses, it is turning RNA into DNA and then back to RNA and then to proteins. So that's kind of the main process here. But let's talk about what the parts of synthesis is. So kind of an interesting thing. The end goal is usually to make proteins, but that's going to be the late phase. And in trying to do this, the virus is going to use the ribosomes that are present in the host cell. Because remember, ribosomes will always have everything to do with proteins. But this is going to happen in an early and late stage. So this slide is still showing us that step 4, that synthesis, that making phase, where we have that early part and a late part. The early part is going to be making more DNA or RNA, depending on what type of virus we have and what type of nucleic acid they have. And then we have the late part of this synthesis, this making section, and that's making more proteins. So again, the virus is going to make proteins using the ribosomes of the host cell. Again, kind of think about going into someone else's kitchen and making them dinner. I know that sounds like a nice thing, and viruses are doing anything but something nice. But they brought some stuff with them in order to make new viruses, but they still have to use their environment that they have now put themselves into, if you want to think of that way, using someone else's kitchen. That leads us into the assembly step. This is step 5, assembly. So this is putting the pieces together. Now that we have built all of the parts, we need to put them together kind of like a set of Legos, if you will. If you have a naked virus, then this step is the last step, and the virus is good to go and ready to take on the world, if you want to think of it that way. If, however, the type of virus is an enveloped virus, then we are going to need to go on to the next step. That's step 6, the last step. But again, step 5 is assembly. It is just building those parts that we just synthesized, putting them together. Step 6 is going to be the release. That is going to be where all of those new viruses that have now been assembled are going to escape the host cell where they were created and then go find other host cells to infect. That is the end-all be-all goal. Now, the number of viruses that can be released by an infected cell is going to vary, and it depends on a few different things. The size of the virus, so for example, the rhinovirus is particularly tiny, so you could probably fit quite a few viruses into a host cell before they are released. Versus Ebola. Ebola is a particularly large virus, so they're going to fill up the host cell a lot faster than something like a rhinovirus. The other thing to take into account is the health of the host cell. This is going to vary whether or not the host, the whole host, is sicker, how far along in the infection they are, and this is going to impact how many viruses those host cells are able to hold and produce, and so that will impact the virus. There are 2 different primary methods for release of the virus from the host cell. The first is cell lysis. This is going to be the most common type of release. It's essentially going to be where the cell, the host cell, ruptures or explodes, kind of like an overfull water balloon. The other type of method for release is budding. This is going to happen with those envelopes or enveloped viruses, where the new viruses are going to bud off of the host cell, taking pieces of the plasma membrane with them. So eventually, this is going to kill the host cell. It's kind of like death by 1,000 cuts versus that cell lysis, which is going to be like an instantaneous, you are too full of these new viruses, and then pop, right? So, this type of budding, though, I do want to point out, we have talked about budding before when we talked about yeast reproduction. This is a completely different process. So before, that was super cute, super adorable how yeast is reproduced. This is viruses, something totally different. It is the new viruses stealing the plasma membrane before going and finding a new host cell to infect. And then, gosh golly, there it is. So you have the viruses being assembled, and then budding off of the plasma membrane. So there it is. From there, we have a kind of overall view of those 6 steps of our reproduction cycle of a virus. Make sure you know these 6 steps, guys. Now, not always does the host cell die in the process of being infected with a virus. And so what happens is sometimes these cells end up getting damaged, and this damage can be kind of specific. So on the left in the picture, we can see cells becoming sticky as well as multinucleated, meaning they have multiple nuclei, so more than one nucleus. What happens is, because the nucleus is going to be the organelle in charge of kind of dictating what's happening in the cell, it's kind of like there's too many cooks in the kitchen, and this mass of sticky cells, if you will, that becomes one giant multinucleated cell can't tell who's in charge. And what happens is it still reproduces, still ends up growing, making new cells or new parts to this mass of cells, and it can become a cancerous tumor. So this is one of the ways that damage to the host cell can cause cancer due to viruses. We'll talk about that a little bit more in part 2. In bacteria, damage to the host cell can look like extra storage spaces. We call these inclusion bodies, but unfortunately that just means that these inclusion bodies won't work properly. And what this does is it just takes up space in the cell, and that doesn't work very well. So even if a cell is able to survive a virus, that doesn't mean that they get off scotch-free. Things can still happen to that cell negatively. So with that, I think that's a good place to stop for Chapter 5, Part 1. Stay tuned for Chapter 5, Part 2. Make sure to be studying, guys, because this unit is very thick. It's a very big unit, and there's a lot of information.